Welcome everyone. Good morning, good evening, good afternoon, depending on where you are in the world today. Thank you so much for joining. A little bit about the logistics of the webinar today. We are recording and we'll be sending this afterwards so you can share with other folks that weren't able to make it today. And if there are any questions, please feel free to add them to the Q&A. We'll try and keep track of it during the call, but then definitely, excuse me, try and look at those at the end and save some time for questions. So today I'm briefly going to set the context for why labs adopt LIMS. And then we're really going to be spending most of our time walking through a live demo using a kind of a fun, fictional, but very realistic scientific scenario. We're actually going to be going sort of interstellar today, but the workflows that you'll see are really similar to what real labs deal with every day. It just allows us to have some generalization to our tools and show you how flexible our LabKey LIMS is. And so before we jump into this demo, I just want to spend a few minutes on who LabKey is and why labs use a LIMS in the first place. So a quick word about LabKey. We've been around for over 20 years, working across life sciences, environmental sciences, mining, agriculture, and more. And what that really means in practice is that we've seen a lot of different lab workflows, regulated and non-regulated, and we design our software to adapt how labs actually work, rather than forcing you into a really rigid process or having to change your SOPs based on the LIMS software that you're using. And so today's demo really aims to reflect that. We've got multiple sample types, evolving workflows over time, and different types of data all living together in a centralized system. And what makes LabKey different is that we're not just a software vendor. We really see ourselves as partners in science. And that means we spend the time to understand how your lab works, help configure those workflows to fit your reality, and really support you all the way through adoption and maintenance. So quickly at a high level, LabKey LIMS is a configurable, low-code, cloud-based platform designed to be easy to use in real labs. So instead of forcing you into these rigid workflows, it really adapts to how your lab actually operates and continues to adapt as your science evolves. One of the biggest challenges we see is data spread across spreadsheets, ELNs, instruments, and shared drives. LabKey helps connect that data so teams aren't constantly reconciling information by hand or spending valuable time and resources looking for data that's needed. And as labs grow or become more regulated, permissions, traceability, and audit readiness are already built into the system, and it's not sort of bolted on later. So it's really a scalable solution as you continue to expand. And so let's make this a little bit more concrete. And in practice, this really starts with registration and uniquely identifying every sample or material that comes into the lab. And that's how you avoid mislabeling and loss as that volume of material comes in to your lab increases. And so from there, LabKey maintains this end-to-end traceability across people, instruments, locations, handoffs. So you always know a sample status and its history. Labs also use LabKey to standardize repeatable workflows. So preparation of samples or materials, testing, review, and potential release. And this gives you clear handoffs and checkpoints without having to micromanage scientists within the lab environment. All of the results are captured within a full scientific context. So we've got the metadata, methods, attachments, any related records. And so that data actually becomes very usable later. And then the payoff is really having the audit readiness. So you don't have to reconstruct what's happened. That record is already there when you need it. And so in today's demo, a research institute has returned from a deep space mission to a planet called Xenon 9. They brought back two categories of samples: environmental samples from different planetary sites and biological specimens from newly discovered organisms. And so the lab's goals here are actually pretty familiar. We want to collect and process those environmental samples, analyze the environmental composition, collect the biological samples, and then extract, sequence, and store that DNA. So across all of this, they really want to answer any of these questions at any time. So where did the sample come from? How was it processed? What data was generated? And today we're going to show you kind of how that works in practice in LabKey. So this is the LabKey LIMS dashboard for our fictional project, Project Xenome. And you can think of this as your lab's home base, the real-time snapshot of what's happening right now in the lab. So across the top, this works the way you'd expect from most modern web applications. You can search for samples, sources, and records globally from here. And you can also switch between projects and access your notifications or any of the system settings. And so the nice thing about our global search is that you really don't have to remember where anything lives. The search is always available for people to be able to find what they need. And so in this Project Xenome project, everything that we're doing today lives kind of within this folder. And folders within LabKey let labs organize the work by study, program, customer, mission, while still being able to use the same underlying system. So if you have multiple studies or teams, you don't have to end up with separate tools, just these separate folder spaces. And on the dashboard of this home base here, we're able to see all of the samples within the system, both samples that we have currently within our lab, but then anything that we have processed. So you don't have to go through and traipse through any audit logs if you need to troubleshoot or understand what happened in the past. And the point here isn't these exact numbers, it's really getting that overview into understanding the shape of the work in the lab. I, as an administrator, have customized all of these, and so this would be likely very different for the individual labs tracking their samples within LIMS. And over here on the right, we can see our notebooks panel, and these are our electronic laboratory notebooks that are directly tied to samples and data within the system, and it's not a separate document living somewhere else. So we'll come back to these a little bit later as we see how that scientific context is captured. For the locations recently added to, this is really our storage activity. And this is showing a live view into which freezer locations were recently used down to that shelf rack or box level. And what I like about this view is that it really connects that digital system back to your physical reality. And so these samples exist somewhere, and the system knows exactly where they are. And then lastly here, our workflow job component. And these are my active jobs that I have been assigned. And this really represents workflows that are currently running, things like biological sample processing or our environmental analysis. And this gives lab managers and teams really immediate visibility into what's in progress, what's due, and what might need attention. And so the last thing here on this overview is our navigation menu. And this is really how our system is organized. And across the top, everything is really set up by what it is, not by the tool that's being used within the system. And this allows people to really easily see themselves within the lab or understand where they need to go to register or add new data. So sources are where samples come from. In this case, we've got collection site and organism. Our sample types, those materials that you want to track. Assays are where your test results live. Storage, obviously, where the samples are physically exist. And our notebooks that work on tracking experiments in prose, as well as our workflow, which allows for labs to track processing or very regulated steps that they want to have on their samples. And so this structure really stays consistent no matter how complex your workflows get. And typically, once our customers understand this structure, the system becomes quite intuitive. And so you're not really hunting for different features, you're really just moving through your science. And so, with that in mind, we're really going to start kind of upstream and look at where our samples came from. So, I want to start at the collection site here. This is all metadata that I added as an administrator that was relevant for my collection site. This would look different for you, very similar to Excel. All of our grids can be sorted and filtered. You can export these out or build charts on them as well. You can also customize the view, so you can have multiple saved views for data that's relevant to you at that time. Individually, for any collection site, we're able to go into this collection site and view all of the samples that are connected with it, that data that we saw on the grid, and then, of course, any notebooks that are referencing this specific collection site. We have a roll-up feature within our LabKey LIMS that allows you to see any assay data associated with samples from this collection site, as well as any workflow jobs that the samples have undergone associated with this collection site. So for this source, this really could easily be patient cohorts, field locations, or manufacturing lots. It's really about sort of the structure that matters to you and how you would want to track that within your system. So if we look a little bit downstream at the environmental samples, one of the things that will look familiar here is sort of the metadata that we've added, as well as the information about the collection site is something that we're able to pull through from our lineage diagrams and understand kind of that full context of these environmental samples. Again, if I go into an individual sample, I'm able to see more metadata about that sample, as well as any notebooks that are referenced. If I look at our lineage diagram, I can actually see where the sample came from and move through this lineage tree. And so this actually shows me all the samples from that collection site we were just looking at, or if there's any samples that have been processed after that. So if it turns out there's a contamination or a deviation in the protocol that puts this at risk, I now have a very good list of samples and results that I might need to modify or remove from our final analysis. And so for each of these as well, I'm getting a similar view into the assays or jobs that might be part of this work. And then lastly, the timeline, which is very important in both regulated environments, but then also as a troubleshooting tool. So if something is abnormal with my data that I've received or not expected, I'm able to go into here and see exactly what has happened to the sample since it's been registered and who did it and when. So in this case, with the sample update, we can see that we made a change to the status and it was me a few days ago. And so each of these environmental samples essentially create these environmental extracts. These extracts also have their own metadata associated with it. They're captured here within storage. I can do the normal things that I would want to in storage. I can move these samples. I can check them in and out. I can export a grid of these samples or even a box view if I need to. And then lastly here, what we want to do with those environmental extracts is actually track the composition analysis. And so these are the results for that composition. I've been able to capture the sample and pull through that collection site, so I'm not having to duplicate data entry here, as well as capture the results of this composition analysis. I have some calculated columns and conditional formatting to highlight data that might need review. And I'm also able to build charts on this data. So I've got two sort of charts set up here to look at that mineral index by collection site or my organic content by the collection site as well. So all of this processing can happen individually by folks within the lab, but really it's much more easily managed when we work through our workflow jobs list. And so if I go into my environmental analysis job, I can actually see that we've got the process environmental samples template that has been designed by an administrator to say, here are the steps that we want you to do when you're processing these environmental samples. I can see in these individual tasks and have those assigned to people. I can see what work has been executed. I can also see all of the samples that have been either inputs or outputs of this job. And then, of course, any assay data that has been associated with it as well. And so, at a high level, this allows me to track the work within the lab much more easily. I'm also able to set deadlines, add myself to a notify list if I want to understand where this is happening in the process. There can be attachments that are added here for images or deviations that happen, or comments can also be added here to be able to collaborate with the people who are executing the work. And then lastly, the other piece of sort of experimentation here is really being able to track the pros of what we're doing within the lab in our electronic laboratory notebook framework. And so I've got our environmental sample analysis as well here, but let's look at the biological sample analysis since we haven't looked at these yet. And here again, I've got a notebook that I generated, and I could have chosen a template here, or I can actually save this as a new template. And we're able to capture high level experiment objectives, the backgrounds. I'm referencing the samples analyzed directly within the system. And so again, there's a sort of data awareness to our electronic laboratory notebook that allows us to at mention or basically copy and paste the samples, sources, any relevant assay data or workflow jobs into sort of a single centralized notebook. All of those referenced items are captured here on the side, and these are all live links to data within the system. I can also go through and paste in images here, or start comments as well to collaborate. Our notebooks have kind of co-authorship, so I can add another author if there are other people working with me on this project. And now Keith would be able to contribute to this ELN as well. I can submit this for review and get this signed off and locked. And once these are locked, I can export both the PDF, but I can also export all of the notebook data at the time of snapshot. So this gives me a very clear picture of what the samples or sources or assay data was at the time of the notebook review for any IND submissions, or maybe in this case, submissions to the government about what we found in our planetary sites. So I'll do take one more step back here and just sort of quickly look at our organisms and DNA samples that we've collected. So from our organisms, we can connect these to collection sites as well as any of the downstream samples. And so again, I've sort of created some conditional formatting to have the risk level of the organism sort of highlighted. We can add images here. If I go into one of these, I can look at that lineage again and kind of see all of the samples that have been generated downstream, as well as any of that assay data and workflow job component. We talked about sort of searching before. So now if I really wanted to search for this organism, I would be able to go in here and copy and paste or even scan if I had that available to me and find that organism as well. Lastly, we want to make sure that it's very easy to find samples within the system based on the context that you have. And so our sample finder tool allows you to search for samples that might have certain properties, but you may not know kind of the list of samples that you need. So maybe we're looking for organisms that are microbes that came from uh, a certain type of biological sample that maybe has a certain amount of material that we want left. So we'll just say is not blank. And that now gives me a list of the samples that we have within our system. I can select these. I can add these to a workflow job. I can choose to act upon them, remove them from storage, generate reports on what assay data is associated with these microbe samples, or export it for further sharing. So with that, I'll kind of move back into the slides here. And so hopefully, you saw this very fictional science that we made up, but kind of the real lab work that might be applied to your lab. And so if you're interested in seeing this mapped to your own workflows, one of the things that LabKey does very uniquely is run these free guided trials. And these guided evaluations basically take on three phases. There's the discovery phase where we're learning about your requirements and kind of mapping the LabKey LIMS to your process. And then we help and configure and demonstrate your processes within our system to let you see kind of what that looks like. And then lastly, kind of pass that on to you to be able to explore the trial on your own, so you can test it out and really get other lab members involved, as well as making sure that it's going to fit with your requirements. So if you're interested in doing something like this, definitely keep an eye out on your email for a link to schedule that meeting, and we can get something set up. So I'll look here kind of at the questions. Feel free to put more questions in the chat or the Q&A, and I'll try and get to answer them in the next couple minutes here. Yeah, so one of the questions is about connecting assay data. So we have kind of that analysis directly associated with the samples. And so the first question is around if there's APIs that connect to LabKey and bring in that data, or do you have to manually enter them in? And the answer for all of our data structures is actually both. So somebody can choose to import data directly within the system. This can be through data in the grid or importing from a file. We also can transform files that are directly instrument generated into a more sort of database friendly tabular format to come in. Or if you have the expertise and the desire to automate this, labs can use one of the many LabKey APIs that we have endpoints for to register or import this data via our APIs. All of those APIs will follow the same permissions that the users have and the same sort of requirements of the data as well. And then the second question is around can AI be used to analyze multiple data sources for that sample? And yeah, so I think what makes LabKey very AI friendly is basically the structured data that we can apply to the system. And so we have actually multiple people pulling data out of LabKey to feed it into AI or ML models to be able to predict something that a human wouldn't normally predict. We're actually looking at tools within our own software that we can help apply kind of AI to, but the machine learning, because those algorithms can be so specific for organizations and sometimes even proprietary, we want to be able to enable teams to pull this data out in a format that's more easily recognized by AI. And yes, so another question, is the system used in a clinical laboratory setting or more for research type labs? It certainly can be in a clinical laboratory setting. We have a number of kind of highly regulated customers tracking samples within our system, and happy to talk to you about your specific requirements here. Is this cloud or on-prem? So mostly this is a cloud-based solution. We do have product tiers that would allow for customers to have LabKey LIMS on-premise as well. And yes, LabKey also has a presence in UK and Europe. And the best way to connect is to follow the links that we have. We have folks in the UK as well that can meet with you. Oftentimes on the West Coast here in the United States, I'm up early to meet with those customers as well. So happy to connect regardless of the time zone you're in. And then maybe the last question here is the free version PHI secure. So I think you're sort of referencing the community edition. The community edition is something that folks can sort of manage on their own. We do not have this LIMS within the community edition. So that might be something for us to kind of follow up on and discuss a little bit more. Okay. With that, thank you guys so much for staying. This was a fun webinar to give in this kind of outer space realm, and looking forward to meeting with you guys to kick off some of these guided trials.